Yes, so we have from Nanobiotix Dr. Matthieu Germain. He joined the company 2004 and is the leading person of the chemistry team at the company.
Thank you for this nice introduction. I will take advantage of the 10 minutes you will need to read this slide to thank the organizer to give me the opportunity to give this talk. So thank you for that. In Nanobiotix, we are a nanomedicine company. Our main product, NBTXR3, is based on hafnium oxide nanoparticle, which are designed to locally enhance energy dose deposition during radiotherapy treatment. We are in clinical phases in 7 indications with this product, and we're waiting our CE mark by the end of the year. But I'm not here to talk about this product. I'm here to talk about a new platform we're launching in Nanobiotix, which is called Redefining, based on a problem we faced as most of people working with a nanoparticle, nanomedicine. When we inject intravenously this kind of product, this therapeutic agent, I would say, not only nanomedicine, we face the same problem. Only a small dose, small part of the dose is responsible for treatment efficacy. Rest of the dose is useless because it's mainly trapped in several organs. One of the main organs involved in the clearance of a therapeutic agent, everybody knows it, it's the liver. It will lower the dose available for subsequent accumulation in other tissue. It could bring some hepatotoxicity. So we're wondering how to modify the bioavailability of therapeutic agent or to redefine benefit ratio playing on the liver. So for us, one of the main problems we face with therapeutic agents is we have to ensure in a single object, a nano object, all the function to ensure treatment efficacy, from biodistribution to tissue diffusion, drug release, and so whatever. So there is a notion of compromise between this physicochemical attribute in the design of the therapeutic agent. And what we want to do here is to uncorrelate the function in 2 separated objects. The first one will take in charge biodistribution, circulation of the second one, which is the therapeutic agent itself. So there is less notion of compromise in these 2 objects, so you could increase several functions in your therapeutic agent, meaning as for nanomedicine, you can play on the level of pegylation to increase cellular uptake of this drug carrier. So our idea is really to prime the body to receive the treatment, or in other words, I would say, keep the liver busy, we said in nano. Because in practice, what we want to do is to design a nanoparticle with defined physicochemical attribute which will reach the liver after intravenous injection. It will saturate physically and transiently the cells involved in the clearance of your therapeutic agent. So when in the second time you will administer your therapeutic agent intravenously, you have less clearance in the liver, so you are more dose bioavailable for subsequent accumulation in the target tissue. And you can have also less hepatotoxicity when there is some hepatotoxicity. At this point, we could say that, yes, there is already some approach which has been developed in this way, even in the 80s, with different objects, but we think we can really improve this kind of approach by designing maybe differently in the nanoparticle responsible for the liver saturation. And we can really adapt this nanoparticle to the needs of different therapeutic agent family. In our platform, we've got several approaches. One for the nanomedicine, where we well know that Kupffer cells are massively involved in the clearance of therapeutic agents, so we'll focus on the saturation of this Kupffer cell. We've got our approach for small molecule, where the main problem is the first-pass metabolism in intestine and liver. Here we are focused on the liver, so we designed some nanoparticle nanoprimer with CYP inhibitors inside to deliver these CYP inhibitors inside the hepatocyte to avoid metabolization of your small molecule. So there is different approach which could be used. Always also in the goal to adapt this approach to the needs of your therapeutic agent. If we represent the total dose administered to a patient with this vial, only a small part will be responsible of the efficacy. The rest is useless because it's trapped in the liver. It will bring some hepatotoxicity. If your product is limited by its efficacy, for the same dose administered, we could increase the efficacy of the treatment. In another way, if the product is limited by its toxicity, we can keep the same efficacy with a lower dose administered to the patient to increase quality of life and treatment persistence, I would say. I just wanted to present you preliminary result we generate on the approach where we want to saturate Kupffer cell using our nanoprimer. For this, we test different kind of nanoparticle. Some are nanoparticle polystyrene, over with hydrogel and liposome. We focus on the liposome finally because we define physicochemical attributes of this liposome, and they're quite interesting because they're well known. So we use finally in the result I will introduce you a liposome of 300 nanometer diameters, highly negatively charged, to saturate the liver. The first point was to see the biodistribution of this liposome for sure. For this, we use in first intention in vivo imaging. We fluorescently label our liposome and we follow its biodistribution after injection. And why do we perform first? It is because we want to know at which time point this nanoprimer will be accumulated in the liver, just to define the delay between the injection of this nanoprimer and the therapeutic agent for further experiments. So we see that after 10 minutes, okay, almost all the dose administered of the nanoprimer is accumulated in the liver, so it helps us to define the window between the two injections. For sure, we perform more quantitative analysis using this nanoprimer by ACPMS with loading of metal oxide nanoparticle inside the liposome. We obtain about 60 to 70% of the dose accumulated, mainly in the liver and 10% in the spleen. Important point, there is no accumulation of our nanoprimer within the lungs or remaining in the blood after injection. Of course, we perform also a preliminary safety study by subacute evaluation of toxicity. For this, we use this nanoprimer and we perform 3 repeated injections of this nanoprimer spaced of 24 hours. And we follow the mice for different parameters that we evaluate 24 hours after the last injection or 7 days after the last injection. And finally, we didn't obtain any modification of these different parameters, body weight evolution, azathalate, total protein, albumin, urea. So for us, there is no sign of subacute toxicity after this 3 repeated dose of our nanoprimer. So we can move on the evaluation of the impact of the nanoprimer of different nanomedicine bioavailability, circulation in blood. For this, we use different kind of nanomedicine. Here I present you data obtained of PLGA nanoparticle of 140 nanometers, fluorescently labeled. When you inject alone this nanoparticle, 1 minute after injection, they are mainly trapped in the liver and the spleen. But if we perform 10 minutes before an injection of this specific liposome, what we obtain is that the nanoparticle, PLGA nanoparticle, continue to circulate on longer time, until 60 minutes for this experiment, inside the blood. An important point, there is no accumulation in the lung or other secondary organ of the MPS. We repeated the experiment with different kind of nanomedicine, always the same kind of result. Important point is that the saturation is transient. If we increase the delay between the 2 injections, the effect is less pronounced, and if we increase drastically the effect, there is no more effect of our nanoprimer on blood circulation and liver trapping of the nanomedicine. Just to show you another example, we've got a collaboration we just started with MIT, with Robert Langer's lab. Everybody knows they're working on different kind of nucleic acid drug delivery system. So we try to evaluate our approach with their nucleic acid delivery system. Here's just an example. We just started, and here we are, the lipidic carrier alone. mainly trapped in liver and spleen. And when we combine a nanoprimer with the same delay, we increase the circulation in the same range of value of this nucleic acid carrier. When we perform ex vivo experiment, you see without the nanoprimer, high dose accumulated inside the liver, and we decrease this dose with the combination of the nanoprimer. And here, the increase in the blood of the circulation time of the nucleic acid carrier. It's ongoing now for transfection experiment to see how we increase efficacy of this nucleic acid delivery system. It's ongoing, but in parallel, we start to perform evaluation the impact of our nanoprimer on drug delivery system. For this, we use the irinotecan-loaded liposome, homemade but very close to Honeyvid, the marketed product. And we perform anti-tumor efficacy experiment on colorectal HT29 tumor model bearing mice. And what we obtain is that when we inject our irinotecan-loaded liposome at a dose of 30 milligram per kilo alone, we've got this curve. And when we combine the nanoprimer to the same dose, we increase the efficacy until the same efficacy to nanomedicine alone of 45 milligram per kilo, meaning that combining on nanoprimer is equivalent to a 50% dose increase of nanomedicine. This was the first experiment, so with a small group of animals. So I don't have overall survival, but we're just finishing a new study with larger group of animals, and I think we'll have quite nice result in terms of overall survival for this animal. So this is where we stand at the moment, and we think really we can modify the way we administer intravenous treatment. And for us, it's not a single product. We can make a platform with this to combine with different nature of therapeutic agents, as I introduced you. For sure, there is a lot of work to do. What is the impact in the complement? At the moment, we only perform in vitro C3, C5 experiment to see the impact of nanoprimer on the complement on human blood. There was no modification, so we are quite happy with that. But there's a lot of study to do, understanding the mechanism for regulatory purpose. All this is ongoing, so we'll see later. Just want to thank the people involved in Nanobiotix within this project, and also Marion, which is in MIT. For sure, also thanks Robert Longer for his support. Everybody in Nanobiotix surrounding our team, people in VoxScan performing the in vivo imaging experiment, and everybody in Nanobiotix. Thank you for your attention.
Thank you very much. Questions to? Yep. Yeah.
Hi, thank you very much for your talk. We're just wondering how confident you are that your nanoprimer is sticking specifically to the Kupffer cells and not also sinusoidal endothelial cells. And if it is 100% only Kupffer cells, do you think there might be a problem? Because endothelial cells can take up particles, especially smaller ones. Therefore, given that endothelial cells are larger, have a higher number than the Kupffer cells, might you have problems there?
I fully agree with you. Yes, we said Kupffer cell it's the concept, but for sure, yeah, we know liver, sinusoidal endothelial cells will be involved in the clearance, and maybe we will have some accumulation of nanoprimer inside. And maybe it's part of the efficacy. That's ongoing to see what is the percentage between the two population of cell. So I will see the result, but maybe that part of the efficacy, I would say. I'm pretty sure of that.
Okay. Very fast or very, very fast? A very fast answer.
It's a very nice sort of revitalization of an old concept, because they did to prolong the circulation time of liposomes, empty liposome injections first to saturate the MPS system. But in your case, with the nanoprimer, if you have a nanomedicine with severely ill patients, immunocompromised, if you have the nanoprimer in your Kupfer cells, and the Kupfer cells is busy with the nanoprimer, then
What is
it's
the impact?
degrading it, maybe slowly. You may have a very long period of immune suppression of the first line of defense. So there could be an increased risk for infections for these patients. Is that a risk?
Yeah, we will saturate the Kupfer cell for, I would say, for a window of 24 hours.
You're sure that the nanoparticles are degraded by then?
The impact on nanomedicine circulation,
Yeah,
if perform delay, it's no more efficient, I would say. But my point is, I think we've got already the answer. There is some product which kill the Kupfer cells. I mean, for example, doxorubicin. If you put doxorubicin inside a Kupfer cell, you will kill Kupfer cells. We use this treatment.
Yeah, but that is different because then you kill,
that was
then
not
you kill,
short.
and then
Yeah, they are replaced. They are replaced, but in, I would say, I don't remember, something like between 4 and 7 days. So you have to replace the Kupfer cell, and during this period of time, your patient is immunosuppressed for Kupfer cells. In our case, yes, there is saturation, but in a shorter time frame, I would say. So I'm quite confident this is not really a problem. There's a lot of products which play on this Kupfer cell and which are used in clinics. So, but we have to ensure this. I fully agree. Yes.
I suggest you have a drink together tonight, okay, to discuss this interesting topic further. So now we come